estimado público, a la defensa de tesis, que Paula decía es un momento de celebración, es un momento para que todos reconozcamos el esfuerzo, el trabajo de doctorado que Paula ha realizado y para que todos disfrutemos del resultado final de todos esos años de trabajo. Los miembros del tribunal, Gabriel Sanpere Baró, Irene y Omar Cornejo. Gabriel, bueno, yo creo que de hecho cada uno de nosotros... ¿Funciona así? Ah, mira. Cada uno de nosotros quizás podemos presentarnos un momento y decir quiénes somos. Yo soy Omar Cornejo, profesor asociado en la Universidad de California, Santa Cruz. Trabajo en la genética de poblaciones de parásitos hospederos, en la evolución de parásitos hospederos. Gabriel.
Jo soc Gabriel Sanpere, treballo. He començat un grup recentment a l'IMIM, on estudiem l'evolució del cervell humà.
Yo soy Irene Hernando Rack, trabajo ahora como Junior Group Leader en el EBMB y trabajo sobre la memoria epigenética de las células y la plasticidad celular.
And with that, we're going to have to change to English because it's the language of the UPF for the defenses. So we are going to be listening to the, oh, yes, there you go, to the talk by Paula called Evolutionary Insights into Primate Gene Regulation and Development of Methods to Study DNA Modifications. With that, let's enjoy it and talk about it later.
Vale, ahora os sentáis. Okay, so first of all, good morning. Thanks everyone for being here, especially the members of the committee for agreeing on evaluating my PhD dissertation, which is entitled Evolutionary Insights into Primate Gene Regulation and Development of Methods to Study DNA Modifications. This is a work which has been conducted at the Institut de Biologia Evolutiva, and it has been co-supervised by Drs. Tomás Márquez Bonet and David de Juan Sopeña. In this presentation, I will first start by introducing the major questions that we address in this thesis. After that, I will read the general objective of my dissertation. Next, I will walk you through the major results that have been obtained in this presentation that are divided into 3 different chapters. And finally, I'm going to read the main conclusions for each of the chapters. So let's start with the introduction. In 1957, Francis Crick drafted the idea on what would be later known as the central dogma of molecular biology. He postulated that the genetic information flows from DNA to RNA to protein, and even though this version and the following versions of this framework provided a molecular basis for the connections between genotype and phenotype, they were not able to address how is it that such molecular pathway is controlled. The answer to this question is gene regulation. In this sense, gene regulation is able to explain how is it that cells, which essentially carry the same genetic information, are able to give rise to such an extraordinary cell diversity, which will turn into different tissues and organs with different functions. Moreover, gene regulation is also able to explain how is it that different individuals are able to respond to different stimuli by regulating genes accordingly. So gene regulation may act upon any of the levels, from DNA to even after a protein has been synthesized. And in this thesis, we're going to focus on the first 2 levels of control, which are the packing and unpacking of the DNA structure, and also the transcription or the gene expression. So most of the changes that involve the alteration of the structure of the DNA are established during development and are maintained as the cells divide in a phenomenon known as epigenetics. So epigenetics, which literally means above or in addition to genetics, has been defined as the study of methodically or potentially meiotically heritable alterations in gene expressions that are not caused by changes in the DNA sequence. There are, however, some other definitions that do not consider the requirement for heritability. And while we have epigenetics, which refers to the study of a particular gene or set of genes, we can distinguish it from epigenomics, which expands this study to all these modifications across the genome. But regardless of the exact definition, the major epigenetic mechanisms that are known to stably alter gene expression are the following. The modification of the histone tails by the addition or removal of chemical groups. Second, the reversible modification of the DNA by the addition or removal of DNA modifications, namely methyl groups. And finally, the regulation of gene expression by small non-coding RNA molecules. In this thesis, I'm going to focus on the first two types of these epigenetic mechanisms, and I will start with histone modifications. So histones are basically proteins that help the packaging of the DNA in what is known as chromatin, and some modifications in the tails of these histones have been associated or can be used in order to establish different transcription states. And although there are now more newer and overall more advantageous methodologies in order to detect these methodologies, the gold standard technique in order to profile this type of modification, it's known as a chromatin immunoprecipitation followed by sequencing, or in short, ChIP-seq. As such, ChIP-seq relies on immunoprecipitation, as you would guess. The histone modifications are cross-linked to the DNA. After that, the DNA is fragmented, and it can be enriched. So the histone modifications we are interested on can be enriched through the use of antibodies, which are specific to these histone modifications. And once we have enriched for this fraction of the DNA, libraries can prepare for sequencing, and after sequences have been done and the analysis have been done of this data, we end up with peaks that will tell us the relative enrichment of these histones, and so the relative position of the histone across the genome. And as I was saying, histone modifications can be used in order to assess transcription state, and we have an example of that. We have here two ChIP-seq profiles from two different histone modifications, the one on the top in green, which is associated with transcription elongation, and the one on the bottom, which is associated with a repressive signal. So if we focus here on this specific gene, we'll see that we see a considerable enrichment on the first of the two histone modifications, the one associated with transcription elongation, but not with the one associated with a repressive signal. As such, this is telling us that most likely this gene is being expressed, at least in this particular cell type, which is being profiled. And then if we go and have a look at the neighboring genes, which are precisely located in the negative strand, we see the reverse pattern. We don't see a lot of enrichment for the modification associated with transcription elongation, so the one on the top, while we see a considerable enrichment of that modification associated with a repressive signal. This is telling us that these two genes most likely are not being expressed, at least in the cell type which is being profiled. And so as I was saying, histone modification can be used in order to characterize different transcriptional states, but also genic elements such as entrons or introns, or other regulatory elements such as promoters or enhancers. In this sense, promoters are usually occur with transcription start sites. These are the places in which the transcription factors and the RNA polymerase II are going to bind in order to start transcription. Moreover, we also have enhancers, which may be distal to the gene that they're regulating, or they can be located within the gene that they're regulating. And basically, they bind to the transcription factors to enhance, and that's the name of enhancer, the transcription of the specific gene that they are regulating. It's important to take into account that enhancers play a key role in driving cell type specific expression. So the combination of all of these modification marks allow us to segment the genome in regions with different functional properties, which are known as regulatory states. And this is going to be important for the first chapter of the result of this thesis. Now I'm going to move on to the second type of epigenetic mechanisms, DNA modifications, on which I will talk today, DNA methylation and DNA hydroxymethylation, and I will start with DNA methylation. So, because it's the modification which is most common in mammals. So when we talk about DNA methylation, basically we're talking about this structure over here, in which we have a methyl group in the 5th carbon of a cytosine, and that's the name 5-methylcytosine, or in short, 5-MC. mostly of the dysmethylation, so we can find this structure in those cytosines that happen to be in a CpG context, meaning in those cytosines that are followed by guanine. And there are, however, some, while most of these are going to be methylated, there are some exceptions of clusters of these CpGs that tend to be unmethylated and collocate with promoters, and these we refer to as CpG islands. In this sense, it has been shown that CpG islands, when they are methylated, there's a link with a long, stable repression of the genes that they are preceding. In this sense, this provides a first link in the levels of methylation and also the levels of gene expression. And although this is true, this is not a general idea that is as general as previously thought. And so this is a summary of basically what I have just said. You will see the dark circles, they represent methylated CpGs, and the white circles unmethylated CpGs. And basically here we can see some CpGs that across a particular genomic region, they are methylated, but we can also find some CpG clusters that are methylated, which are CpG islands, and they can be locating with promoters or in other regions of the genome. So there are basically two methods, or I define that there are two methods in order to detect DNA methylation at the position level. These are conversion-based method, one of them, in which basically here what we do is we're going to convert or treat the DNA through either chemically or enzymatic agents. And so the differentially methylated or unmethylated positions are going to be differentially converted. And after profiling this treated DNA, we are going to be able to profile them and that way know which of the cytosines are methylated to the ones that are not. So there are basically two ways in which this DNA can be converted, either using a chemical agent, the most famous one is bisulfite, or now more recently with different combinations of enzymes. I will talk about it in the last chapter of the results. And this treated DNA can be then profiled either using microarrays or next-generation sequencing. Another way in which we can profile DNA methylation at the position level is through the use of long-read methodologies, namely PacBio, or here in this precise example, Oxford Nanopore Technologies. So the best advantage, or the main advantage of these technologies, is that they allow to virtually assess any type of DNA modification as long as you have the methodologies able, or the models, which are trained in order to detect such modifications. And as I was saying, I have used all of these methodologies in the different chapters of the thesis. Now I'm going to go to talk about the DNA hydroxymethylation, which is in fact related to DNA methylation, as it is the first intermediate during the active demethylation of the cytosine group. And if we compare the two structures, basically we can see here we have the methylcytosine and here the 5-HMC. So basically we have here an hydroxy group, and thus it receives the name of 5-hydroxymethylcytosine, or this time for short, and in order to attenuate bit of confusion, 5-HMC. So about the key features of this structure, we can highlight that they are abundant in pluripotent stem cells and also in brain-related cells. In this sense, those disturbances that have been found in these in the levels of 5-HMC have been associated with either psychiatric or neurological disorders. Moreover, this modification is also associated with enhancer marks and also in gene expression in gene bodies, which is independent of 5-MC, so methylation. And regarding the way that they are profiled, it's a similar method to the ones I explained before that relies on bisulfite as a chemical method in order to convert this DNA. So we can say that the last 10-15 years we're living with epigenetic revolution in which a lot of big efforts are being led by different projects and to generate different types of regulatory data, which include epigenetic data, in order to profile a lot of many things. So different human cell types and tissue types, even at the single cell level, also at different conditions, and also even at different developmental stages. So at this point, we really need to focus and pay attention on those regulatory elements that are actually giving us information that could be meaningful for the biology of the human biology to those that are just adding variation. And in this sense, comparative studies could be a useful tool to implement this. However, most of the standard animal models only provide limited information, given that they are relatively distant from humans. As such, one could think that primates is a good way to study these, the implications of such regulatory elements, so through the use of primate comparative studies. However, also partly limited to the absence of good quality references for most of these non-human primates, not a lot of epigenetic data has been generated in the last decade. Moreover, another layer of complexity is added when taking into account that for these interspecies comparisons, we need the same cell type in order to profile the different conditions or in the different species. And this is, most of the cases, very difficult to obtain, given that most of these non-human primates are endangered. And so taking all of that into account, we define the following objective for the thesis, the general objective, to be the study of gene regulation from an evolutionary perspective with a particular focus on DNA modifications. I have looked at this from different perspectives and also using different technologies. And now I'm going to go to present you the result of this thesis. So as I just mentioned before, we have divided the results of this thesis in 3 different chapters. In the one, we have had a look at the characterization of different regulatory elements through the use of a comparative primate study. In the second one, we have assessed a structurally complex chromosome. That's the case of the Y human chromosome. And finally, in the third and last chapter of this thesis, we proposed a methodology in order to detect methylation and hydroxymethylation. So I'm going to start by the first chapter. The results of which have been included in this article, which has a long name. Epigenomic profiling of primate lymphoblastoid cell lines reveals the evolutionary patterns of epigenetic activities in gene regulatory architectures. So the main motivation of this work was to characterize in a comprehensive manner the regulatory elements present in primates. And as you will see, this is quite an extensive project that today I will only focus on the analysis that I have performed myself, which have the following objectives. However, in order to understand my analysis, I will first introduce the data which was generated and also some of the basic processing of this data. And as I was saying, the particular objectives that I have in this project were to assess the functional implications of different regulatory elements and to inspect the particularities of human-specific regulatory elements. So basically, this is a summary of our study design. We used LCLs from 5 different primate species: human, chimpanzee, gorilla, orangutan, and macaque, to replicates. You will see that in the case of one of the human replicates, the data was already available, so we didn't have to generate it ourselves. For all the other data, it was generated from the other co-first author of the paper. And basically, it included expression data through the use of RNA-seq, accessibility data through the use of ATAC-seq, 5 types of histone modifications through the use of ChIP-seq, methylation data, and also whole genome sequencing data for the remaining cell types. However, in order to annotate these regulatory elements, we only made use of the ATAC-seq data and the ChIP-seq data from the 5 different histone modifications. So in order to annotate these regulatory elements, as I just mentioned, we only made use of the ATAC-seq data and the histone modification data from ChIP-seq in order to define basically 2 types of regulatory elements, promoters and enhancers. You will see them here. Allow me to zoom in. So basically, we have promoters and enhancers with 3 different types of activities for each: strong, poised, and weak. So that way, we have a classification that has 6 types of components. So once all these regulatory elements had been defined in our LCLs, then using the relative position to the gene and also available 3D data, these regulatory elements were associated with different genes. And so we ended up with a proportion of regulatory elements being associated to different type of genes, to other regulatory elements that we could not assign to any gene. They were called orphan genes, and they were not further used in the analysis. And so apart from having this definition that I just mentioned that's based on the epigenetic state, each of the regulatory components, the regulatory elements, were also classified according to their interaction with the gene that they had been associated to. So in this case, regulatory elements that were located in the promoter region were defined as genic promoters. Those enhancers or regulatory elements that were located within the gene, they were referred to as intragenic enhancers. Those regulatory elements that were placed somewhere distal to the gene were referred to as proximal enhancers. And finally, those regulatory elements that were either interacting with other enhancers or with other promoters through the use of 3D data were referred to as enhancer interacting enhancers or promoter interacting enhancers. So you will see that here we have a classification of 6 possibilities, and depending on the regulatory component, we would classify its regulatory element in 3 different categories. So this adds to a total of 30 different combinations in which a regulatory element can be classified. So this is where my proper analysis started. So basically, at this point, we focus on 2 types of regulatory elements: those that were fully conserved in the 5 species of primates that we were considering in the study, and also to those that were specific to one specific species. And as I was saying, we had 30 different combinations of regulatory elements, and we restricted our analysis to those regulatory element combinations that were overrepresented over each component. And so it's here important to highlight that only one type of this combination popped up to be significant in terms of the species specificity, which was the one that you see over here. So those that were human-specific, that were weak enhancers, and that were located within the genes, so intragenic enhancers. And I will be, since they are going to be important for this thesis, I will going to be abbreviating them by putting this. Okay, so once we knew what we wanted to do with these regulatory elements, we designed a set of different analyses in order to inspect more or less which was the functions of the genes associated with these regulatory elements. And so on the one hand, we performed functional enrichment, then expression pattern analysis through the use of the GTEx data, and finally, we also assessed the tissue specificity for each genes. So if we start with the functional enrichment analysis, basically, this is the results that we get, and I know they can be a little bit complex to understand at first. So basically, the circle of the size of the circle is proportional to the fraction of genes that are represented in each group. Here on the y-axis, we see the functional enrichment functions, and here on the x-axis, we have the different regulatory element combinations. So if we zoom in a little bit here, basically, these were the ones that were statistically significant, in which here, the first layer, we have the type of component, that we only have those that are genic promoters of intragenic enhancers. In the second row, we have either one promoter that it happens to be strong, and all of these in blue enhancers. We have the weak, strong, or poised. And finally, here in the bottom row, we have in purple those regulatory elements that were fully conserved in the 5 species, and here in pink, those that were human-specific. So if you remember the human-specific weak intragenic enhancer that I was telling you about before, so it would be represented here, because it's human-specific, weak intragenic, weak enhancer, and intragenic enhancer. And so now we can maybe have a look at the results. So the going to the strong conserved activities, basically most of these activities had to do with general functions that comprise cell cycle or metabolism. In the case of the strong enhancers, some others were also related to functions that are specific to the cell type, which is something that we expect from enhancers. Then if we went to the poised or weak conserved activities, in terms of the poised one, most of them were associated with cell development and differentiation. In the case of the weak enhancers, they were more general, also as expected. And it's interesting that in both cases, they were associated with functions related to the brain, which is not the cell type that we are working with. So this was interesting. And then, even more curious, when we went to have a look at the enrichment for the human-specific weak intragenic enhancers, only 2 functions, and both of them were associated with brain-related functions, were significant. So here, what we see is that weak and poised activities are enriching functions not related to LCLs. Next, I'm going to present you the result regarding the analysis on the expression in the different tissues and also the tissue specificity. So these are the results in which basically we assess the expression through the use of the different tissues as they are included in the GTEx. Basically here on the x-axis you see the same colors that I explained before, and here on the y-axis the different tissues, and the color on the matrix represent the relative expression. The darker the blue, the more expressed, the darker the red, the least expressed. So now that we are familiar with the plot, if we focus on the strong conserved activities, we see that the LCLs, which remember are the cell types that we are working on, show the highest of the expressions, which is something that we would be expecting, right? But for here, all these cell types show minimal expression, and also the brain, that remember popped out as significant in the weak intragenic enhancers that were conserved before. So now if we are to plot the distribution of this expression, we see the same thing that I was explaining you, but with the real numbers. So LCL show the highest expression, and brain minimal expression, with always stays this high expression, but we know that this is a high expressing tissue, and whole blood with minimal expression. And in terms of the expression, the tissue specificity, these genes are mostly expressed, across many different tissues, so they have a high expression breadth. Now, if we focus on the other types of elements, so poised and weak conserved activities, and also the human-specific ones, we see what we would be expecting. So LCLs show minimal expression in these cases, and that's nice. And also falling in line with what we just saw in the functional enrichment, we see now here that the brain show a higher expression. And again, we are working with LCLs, and they are not any way related to brain. So once again, if we plot the distribution of the expression frequencies or values, we see that the LCL show a small or minimal expression, while brain show the highest expression, one of the highest, together with testes, and altogether they show this group higher tissue-specific expression. So as a summary, here we see that weak and poised activities show high expression patterns in tissues not related to LCLs and higher tissue-specific expression. So at this point, we were really intrigued by these elements, not the human-specific ones. And we went and had a look at the manually checked which were the genes that were harboring these elements. And we found out that most of these genes are candidates of genes that have been subjected to different evolutionary forces. We have, for instance, FOXP2, which is a known introgress desert, or CFTR, which is linked to cystic fibrosis. So at this point, we wanted to have a look at them in a little bit more detail. And so for that, we designed two different sets of analyses. On the one hand, we studied the co-occurrence of these elements with signals of selection or acceleration, positive selection or acceleration, and also we also wanted to test whether these elements, apart from being human-specific, also contain some human-specific changes in the DNA sequence. So regarding the first of the two analyses, we found out that the genes that had human-specific weak intragenic enhancers, 40% of them also overlap with the genes with signals of either positive selection or acceleration, and it was statistically significant. We also show this in this hierarchical or nested analysis, in which we show that the intragenic enhancers are the only components that are enriched in this category. Also, out of the intragenic enhancers, only those that are weak are statistically enriched in this type of signals. And finally, out of the intragenic weak enhancers, only those that are human-specific are enriched in this category. So regarding the second analysis, we collected almost 3 million positions that were fixed in humans and different from a variant in different from a variant also fixed in the remaining 4 primate species. So these are called human-specific human sequence nucleotide changes. And so we also had a look at them, and we found out that the density of these type of changes was higher in human-specific weak intragenic enhancers that you would expect in the conserved analogs, in the other groups of analogs. Also, we found out that 3 of these human-specific weak intragenic enhancers show more of these changes in the DNA than expected by random, and in all of these cases, these were genes that were at some extent related or associated with brain-related functions. And finally, we also assessed whether the co-occurrence of these features was also enriched But, well, in this case, only 10% of the genes that had human single nucleotide changes also had signals of selection and acceleration. This is not statistically significant, so these are not enriched, meaning that the two features are not mutually conditioned. So as a summary of this last set of analyses, we show that human-specific weak androgenic enhancers target neuro-related brain-specific genes that are associated with signals of positive selection and acceleration and excess of human single nucleotide changes, although both of these features are not mutually conditioned. Okay, so now I will go to the second chapter of the results. So the results of this thesis, this chapter are included in a manuscript that is under revision, which is entitled Y Chromosome Sequence and Epigenomic Reconstruction Across Human Populations. And although I have led this project, today I will be only focusing on the set of analyses that I have performed myself, and which had a double motivation. So on the one hand, we wanted to assess a novel enrichment technique in order to enrich for an structurally complex chromosome, the Y chromosome. And on the other, we also wanted to assess the methylation landscape across different Y chromosome populations. So basically here, you see a summary of all of these analyses. Basically, we took 7 types of LCLs from 7 different human groups that included Africa, Eurasia, and the Americas. Only 1 replicate per each, and we performed the enrichment of the Y chromosome through chromosome sorting, which is a methodology that physically isolates the chromosome of interest, in our case the Y chromosome, through flow sorting. Moreover, we also wanted to assess the feasibility of using an adaptive sampling, which is an enrichment technique that is already implemented in nanopore. So for one of the LCLs, we also use or try this adaptive sampling technique. After that, all the data was generated using nanopore data, and you have here a representation of all the analysis that we have performed. But today, I will only focus on the one, as I was saying, that I performed myself, that have to do with the method comparison and also the DNA methylation. So regarding the method comparison, we showed for the first time that adaptive sampling is a valid tool in order to enrich for a structurally complex chromosome. That's the case of the Y chromosome. And you can see in that plot over here. So we did this into sequencing batches, but I'm only showing you one. The other is exactly the same. Basically, what we can see is that when the DNA fragment passes through the nanopore and it belongs to the Y chromosome, it is, in most of the cases, enriched for sequencing. So that's why we see this big chunk of green. But for all the other chromosomes, once the DNA fragment that belongs to these other chromosomes passes through the nanopore, it's not recognized as a sequence to be enriched, and thus it is ejected and rejected from the pore. And so this is why we see the biggest proportion in red. However, we also have to take into account that the still chromosome sorting, which was the previous technology that relies on flow sorting, still outperforms adaptive sampling in terms of enrichment factor. However, one thing that we have to take into account here is that for doing adaptive sampling, you need far less starting DNA, and so it may be a better idea to follow in some experimental setups, a better methodology to follow, sorry. And so now I will go to the results regarding the DNA methylation across the different populations. But before I do that, I'm going to first introduce some basics about the structure of the Y chromosome. So basically, what you have here is the Y chromosome. Over half of it is made out of heterochromatin, which is not even resolved in the HG38 human assembly for the Y chromosome. And the remaining part belongs to the chromatin, which is separated into different sequence classes, of which I will focus on 2 because of their distinct behavior. We have, on the one hand, the X-degenerate, which harbors the relics of the ancient chromosomes from which the Y and the X chromosomes co-evolved. It contains single copies genes and also pseudogenes that have their homologs in the Y chromosome. And finally, it shows wide expression of these genes, show wide expression. If now we go to the ampliconic sequence that is colored in yellow here, we see that this is essentially made out of repeats with high similarity between them. It contains many protein-coded genes, but the expression is mostly limited or mostly testis-specific. So now that we know a little bit more the structure of the Y chromosome, I will go and explain the DNA methylation results. So DNA methylation was called using nanopore, and we only kept those positions with a mean coverage of 4x. Here, it's important to take into account that given that we only have one replicate for each of the haplogroups, our results are limited, and so the generalization of our findings is limited too, but still. So basically, this is one of the first results that we see. We see a consistent methylation patterns along the Y chromosome and across the populations. So basically, here you have sliding windows across the chromatin region of the Y chromosome. Here you can see the sequence classes, the genes in the regions, and the strand they belong to. And here the methylation values that go from 0 when they are not methylated, or 1 when they are 100% methylated. So as I was saying, we see some consistent methylation patterns. If we focus on the X-degenerate class, we see that most of, as I was saying, consistency in methylation. But if we focus on the ampliconic region, so remember, this was made of essentially repeats. We see some consistent patterns here, but some others we see a lot of variability. At this point, we also wanted to have a look at the methylation landscape in the gene annotation, also comparing the X-degenerate and the ampliconic regions. So we focus first on the X-degenerate. So remember, this is a region that harbors genes which are expressed in a lot of tissues, so we expect to find expression in LCLs too. If we focus on these genic elements that are closest to the start of the gene, we find lower levels of methylation, which would make sense with these genes being expressed in LCLs. And also, accordingly, we find that most of the CPGs in the remaining regions of this genic structure are methylated. Now, if we focus on the ampliconic region, so these were, remember, these proteins are mostly specific to testes in terms of expression, so we would not expect them to have them expressed here. We find that the genic elements that are closest to the start of the gene are mostly methylated, whereas the remaining CPG sites show more variable or even relaxed methylation values. So at this point, and even though we lack replicates, we were intrigued to see whether we could find some regions that show consistent differences in methylation across haplogroups. And for that, we computed variability as the standard deviation of the methylation across the haplogroups, and found one particular region that show that one of the haplogroups show consistent lowest methylation levels. So when we went and had a look at it, we realized there were some genes here, and so when we then went and had a look at the expression at the CPG islands that are before of the start of this gene, we found out that most of the methylation is constrained. So what this is reflecting is that we may have some methylation differences across different regions of the genome, but in key genomic features, such as the start of the gene, methylation values in general are more constrained. So taken together, here we see that nanopore can be used to study methylation across different cell lines, and it can be helpful for the study of challenging genomic regions. And now we go to the third chapter of the thesis, which basically at this point, we already knew that nanopore could be used in order to detect methylation, but we also wanted to extend this possibility to the study of other modifications. That's the case of 5-hydroxymethylation that if you remember, I explained a little bit in the introduction. But however, in order to be able to detect these modifications through the use of nanopore, we need high-quality data or true sets that can be used in order to train the models in nanopore. And this was basically the motivation for the third chapter of this thesis, the results of which are included in the manuscript in preparation, EO8-seq and SEM-seq, differential detection of methylation and hydroxymethylation through enzymatic deamination. Of note, given the novelties of the methodologies here described, a European patent has been submitted for these two methodologies. And the main objectives of this last chapter were to develop an experimental methodology for the enzymatic whole genome profiling of hydroxymethylation and also for the identification of methylation. So as I was saying before, this chapter arises for the need to have good quality data that profile 5-HMC in order to train models in nanopore. But what's the state of the art of the methodologies for detecting DNA modification? So basically, the detection of methylation relies on bisulfite sequencing, or it's called the standard, BS-seq, but bisulfite sequencing does not discriminate between methylation and hydroxymethylation. On the other hand, for the detection of hydroxymethylation, we mostly rely on oxobisulfite sequencing that needs of two different reactions to take place. One, which would be doing bisulfite sequencing, and also that it's doing a bisulfite sequencing experiment preceded by oxidation. But the problem that all of these methodologies that rely on bisulfite sequencing is that bisulfite-dependent treatments are harmful for the DNA, and thus complicates the generation of high-quality data for 5-HMC. In this regard, as I was also mentioning in the introduction of this thesis, some other methodologies have been available that instead of using bisulfite, they use different enzymatic enzymes in order to modify the DNA. That's the case of EM-seq, enzymatic methyl-seq. So that would be a good alternative for generating this high-quality data, but we need it for the 5-HMC. And although some methods have been proposed that rely on EM-seq in order to profile 5-HMC, the main limitation is that they are only locus-specific, so they would not allow us to inspect this modification across the whole genome. So by having a look at all of this, here we propose an adaptation of EM-seq methodology for the genome-wide specific detection of 5-HMC. So in short, this is the methodology followed by EM-seq, which is commercially available. First, we have an initial step of library preparation. Then the DNA is converted through the use of different enzymes. It's important to take into account that at this point, after the amination, DNA will become single-stranded. This is something that we have to take into account. And after the DNA has been treated or converted, the library is amplified and it's ready for being sequenced. So by having a look at this, which is the proposed methodology in EM-seq, what we propose in our methodology is to change these two steps. So basically here we do a modification of the enzymes that are added in conversion, during conversion, and then we propose to carry out a library preparation that relies on single-strand DNA in order to generate these libraries. And to this combination of the two already existing methodologies, in fact, we refer to as enzymatic hydroxymethyl-seq, or EOH-seq for short. And this allowed for the specific identification of 5-HMC. Moreover, by slightly modifying this protocol, we are also able to detect jointly methylation and hydroxymethylation. So to this second approach, we call single-stranded enzymatic methyl-seq, or SEM-seq, which would be essentially analogous to doing EM-seq, but with a single-stranded library preparation protocol. So at this point, we wanted to assess the performance of these two methodologies, and for that, we took a hippocampus DNA from male donor and performed these two sets of experiments in 3 replicates. And for each of them, we also added 3 additional controls that I will mention a few slides later. So the first result is here. You should see some bars, but it's OK. That we are able to detect this modification in both type of experiments at the whole genome level. Moreover, by using EOH-seq, we're able to specifically detect hydroxymethylation. And while if we use SEM-seq, we are able to jointly detect methylation and hydroxymethylation. However, by using the combination of the two, we can specifically detect methylation levels. And now about the 3 internal controls that we use in order to assess the performance of the 2 methodologies. As I was saying, we use 3 different controls: an unmethylated lambda, DNA, CpG methylated plasmid, and a synthetic oligo, which was 100% hydroxymethylated. So regarding the first of the three controls, since this is a DNA fragment that it's not methylated, and our two methodologies consist on the identification of different modifications, we don't expect to see any in this plasmid. And this is what we find, so no modification whatsoever in the two experiments. In terms of the plasmid, which was solely methylated at the CpG level, we expect to find it in this in the SEM6, since it's able to detect 5-MC, but only in the CpG regions. And basically, this is what we see, almost 100% conversion rates here, and almost marginal in EO8-seq. Finally, as for the synthetic 5-HMC oligo, since the two methodologies are able to detect, in theory, 5-HMC, we would expect to have conversion rates in the two experiments. And while this is true for SEM-seq, we find much lower conversion rates for the EO8-seq experiment in this synthetic oligo. So at this point, we didn't like this, and we went and had a look at the specific, at what was going on in this oligo, and what we found out, and here some points are missing, but that's okay, is that depending on the nucleotide that is preceding the modified base in this 100% hydroxymethylated oligo, we have different affinities of the enzyme. So for instance, if the 5-HMC is preceded by A or G, we see that there's higher conversion rates, and if it's preceded by cytosine or thymines, the conversion rates in general are lower. So at this point, we are still investigating to know what's going on, and if the problem is that we are using a control that is not good enough, or that we have to maybe adapt or adjust a little bit more the conditions of our experiment, or of our methodology, sorry. So overall, here we see that EO8-seq and SEM-seq seem to be two promising methodologies for the identification of methylation and hydroxymethylation at the whole genome level. And now, with the permission of the tribunal, I will proceed with reading the conclusions of this thesis. In the first chapter, we have seen that poised and weak enhancer activities may carry information about gene regulation in unrelated cell types, and that functionally relevant regulatory innovations that appear in a given cell type suggest that they could be echoed as weaker activity signals in different cell types. Moreover, we have also shown that these elements that are specific to humans, so the human-specific weak introgenic enhancers, coupled with signals of positive selection and human acceleration, suggested that they could have a role in human adaptation. As for the second project, we have assessed the performance of different methods for the enrichment of a structurally complex human Y chromosome, and we have shown that adaptive sampling is a cost-effective alternative for the enrichment of this chromosome. Moreover, we have characterized the methylome landscape in human Y chromosomes, and we have revealed characteristic methylation patterns in the different sequence classes that are contained in the Y chromosome. Finally, we have developed, in the third chapter, we have developed EO8-seq, which is an experimental methodology for the enzymatic whole genome profiling of hydroxymethylation. And when combining EO8-seq with SEM-seq, which is specific for the joint detection of methylation and hydroxymethylation, it allows for the retrieval of specific methylation values. So thank you very much for your attention, and I'm happy to take any questions.
Very well. This is a good time for doctors in the audience who want to ask questions to Paula or have comments. Tomás? Absolutely.
Oh, thanks so much. So again, we have here not only academics, but also family members. So if that's okay, I'm going to just give my speech in Catalan, if that's okay. Paula, jo, òbviament, després li passaré al David, però tinc poques coses a dir, entre altres coses perquè és el teu dia i no es tracta de que nosaltres parlem, sinó que ho facis tu. Però jo et vull agrair, òbviament, que fa uns anys, ja fa bastants anys, del grup de l'Òscar Lau vinguessis en el nostre laboratori i que ho fessis per agafar un tema que no és fàcil. És a dir, el treball que tots veieu aquí s'explica en 45 minuts, però són coses molt complexes hi ha hagut moments molt durs, perquè aquestes coses, aquest tipus de recerca doncs, a vegades surt, a vegades no surt, a vegades és interpretable, a vegades ens enfonsem, a vegades ens recuperem, i tu clarament doncs, te n'has sortit molt bé, com jo crec que és evident. A més, em fa molta il·lusió, a més, perquè tens la persona al costat, que és la Irene Hernando, que va ser la persona en el meu laboratori que va començar aquesta línia de recerca. I de la Irene va saltar la Raquel, que està aquí amb una criatura no sé on, si no ha hagut de marxar. La Raquel ha passat al Luis, amb el David, ara la Maria, i tots vosaltres heu agafat una idea i l'heu portada molt més enllà. I jo, clar, ara que ja vaig veient passar estudiants, em fa molt de goig veure com una idea que pot desenvolupar-se de moltes maneres, tots vosaltres la feu portar fins a uns límits que mereixen de sobres tenir el doctorat i fer el que vulguis. Ara no sé què et portarà la vida, perquè això ara és una porta oberta, però escolta'm, en tot cas et desitjo molta sort i moltes gràcies.
Gràcies, Tomàs.
Hola.
Hola.
I will do it in Spanish, because my Catalan is not even real. Bueno, enhorabuena y sobre todo quería darte las gracias por todo el compromiso, la fortaleza y el talento que has puesto en este trabajo, que no ha sido nada fácil. Tuviste el valor de coger un proyecto y unas líneas que involucran una parte experimental y otra computacional, que ya es mucho, y llevarlo a cabo en unas condiciones que, como todos sabemos, han sido particularmente difíciles, especialmente la parte experimental. Y bueno, gracias a esto, a tu compromiso y a todo lo que has puesto en esto, estamos hoy aquí. O sea, que nada, muchas gracias y enhorabuena. Solo esto.
Gracias.
If there is anybody else who would like to ask any questions to Paula? Okay, then we'll have the tribunal now coming to ask some questions. Irene, do you want to start?
Well, también me gustaría primeramente dar la enhorabuena a Paula, a Tomás y a David. Realmente me ha gustado mucho leer la tesis y es genial ver cómo todas estas ideas de la epigenómica comparativa siguen el legado en el laboratorio. Me ha encantado.
Gracias.
So now let's switch to English, and I have some specific questions for you. So for the first part of the thesis, I was wondering, I think it's in figure 1 and 2, at the beginning you have plenty of enhancers, like hundreds of thousands, right? But then when you focus and you do this orthologous comparison between the 5 species, then your data set becomes very narrow. You are only looking at around hundreds of enhancers. Is that right?
Yes.
So I was wondering if you have excluded most of the enhancers because they tend not to be inside the gene regions, right? Have you considered, for example, to expand the coordinates of the orthologous regions to try to capture a higher amount of enhancers, for example?
So you mean for defining the regulatory elements that were orthologous among the 5 species, right?
Exactly. I think there you lose most of your data.
So yeah, so actually this is something that was done by the other co-first authors. So I explained from where the regulatory elements had been already annotated. I think, and from the data, they explored also expanding the coordinates, but
yeah, maybe
this
didn't
data
work.
that was enough.
Okay, no worries. Then, well, an interesting finding and also a provocative one is like you find some epigenetic marks in your cell lines that may have retain some information from other tissues.
Yes.
How do you imagine that this happen? Do these marks will be acquired during development? And also if you have checked other data sets of, for example, human ES cells or iPS cells to see if these marks, they are already there.
So this is the first thing that we wanted to check, actually, whether these human-specific weak heterogeneity enhancers, we could find some marks also in the brain, no? And to see whether they were stronger signals. But the problem is that with most of the data that's already available now, it's difficult. So we did it. So in short, we did it, but I think we need to fully address doing like the experiments almost all over again and using all the different primates because so for the data that's already available, either it's not a lot of data and you cannot annotate the way that Raquel did with the same histone modifications and to obtain the same type of elements, because at the end, weak enhancers are a type of element that it's difficult to annotate or characterize. And either we don't have for all the primates, or either we don't have, we don't have all the modifications for the primates, or we have a specific annotation, but only for humans. So it's difficult to do the comparison across species.
But maybe you could just check human ear cells and just check if the enhancer is active during early development, or if you have already some marks there.
So we checked actually for chromatin states that are already available for different cell types, and nothing was super promising. But again, the data, we couldn't rely really much, and the comparison was not that straightforward.
So do you mean like the signal wasn't there?
Yeah.
Okay. Okay, that's interesting too.
Yes.
Right, thank you. And then, yes, you saw that some of these human intragenic enhancers are associated with genes under positive selection. Have you checked for the presence of a specific transcription factor binding sites, or?
No, this is something that we haven't checked, but it would be, definitely interesting because at the end, there's something going on here. But you mean in the regulatory element itself?
Yeah, in the new ones. So the ones that are only human-specific, so that could explain, for example, that's why you have a new regulatory element there.
So we didn't, actually, that's a good idea. We didn't have a look at that. We only focus on the changes that were human-specific, which was the last part with the...
Yeah, those ones.
Yeah. But no, we didn't do that.
And more like an open question about this chapter is, how do you think these human-specific SNPs or specific mutations, how would you test them to see if they are actually relevant for the human traits? Because this is one of your ideas, right? That these SNPs and these enhancers will be related to some human-specific features, like in the brain development or in brain.
I would do, basically, now that it's very trendy, try to do CRISPR and see whether if we basically add the mutation that we see or the change in the DNA, we see difference in the regulation or the expression of these genes.
Okay. Thank you. For the chapter 2, I was curious. I mean, it's more like a technical question. I'm not sure if you know, but I was curious when I was reading if you know how this adaptive sorting is performed. Do you know if it's based on a probe, a pull down using a DNA-specific probe, or is it using like a specific primers that somehow?
You mean adaptive sampling?
The adaptive sampling.
Sorry. This is nice because I did that. So basically, Nanopore allows you to basically give a sequence that it can be FASTA or BAM. And so you give, in our case, we gave the sequence of the Y chromosome that it's resolved. And basically, when the DNA is passing through the pore, it basically checks real time if that matches with the sequence that you gave. And so if it does, it's when it's enriched for sequencing. And so that's why in the figure, when the DNA that's going through the pore doesn't match to the sequence that you gave, it basically ejects the pore.
That's very interesting. So you don't have any pre-amplification?
No. And that's the good stuff. And that's why we don't need as much DNA to do this experiment as what we need for chromosome sorting, because at the end, you need a lot of cells in order to physically sort the chromosome of interest. So that's actually really cool that we are able to do that with just a couple million cells.
Yeah, that's really nice. Thank you. Then when you look at the methylation of the Y chromosome, I think at some point you look at the CPG islands. Are you using like annotations from some UCSC or other databases? Have you thought about using your own data, because you have such a nice data set, you have DNA methylation and DNA sequence, right?
Yeah.
To try to call new CPG islands, for example, and also on the same lines, if you had tried also to call new active copies of retroelements, for example, that could explain the variability between individuals, or like use your own data to generate new annotations and new maybe active copies of retroelements.
Yeah, so actually we didn't have a look at that because at this point there was a lot of data and we didn't know what to do with that. And so we basically limited our analysis to nanopolish, and so we didn't use data we generated ourselves. And it would have been nice to look at the effect of the methylation of retrotransposons. And I think that's something that they have done in the latest T2T Y chromosome paper that came out just after we kind of published this. And yeah, but it would have been interesting, but we didn't do it ourselves. We basically restricted our analysis to studying the sequence classes because since we didn't have replicates for the haplogroups, assessing variability among the haplogroups was not a real possibility, and so we, as I was showing here, only limited the analysis to the different sequences classes, and that's it.
I see. Okay, thank you. And just the last chapter, I really like the technique. So
Thanks.
yeah, congratulations. At some point, you see, with your oligo, that's your hydroxy levels, they go only up to 70 to 80%.
Yes.
And you hypothesize that this could be an efficient conversion by BGT, right? Mm-hmm. I was wondering if you have any alternative hypotheses for that, and also if you have considered, or if it's known, the ApoVec could react with your glucosylated 5-hydroxymethyl-cytosine, or if you could maybe, or if you have tried to titrate ApoVec concentration or things like that.
So the problem here is that we have used a kit that is commercially available, so EM-seq, and we have basically modified it to generate our own methodology, which is the one that I'm showing here. So we don't really know the concentrations of the enzymes because that's a commercial kit and they don't want you to know. So we haven't tried this thing with trying different conditions of ApoVec. The problem is that we do see 100% hydroxymethylation levels in the other experiment. So I think that the ApoVec is working. So the problem is, my theory is that the control that we are using, since it's 100% hydroxymethylated and half of the positions are cis, I think it's too concentrated. And what you don't see is, because since there are that many modifications, the enzyme is not able to efficiently convert the modification. My hypothesis is, what other papers do that I find tricky, is they only use a oligo with one position that is hydroxymethylated. And then they say, oh, this methodology works 100% of the time. Well, you have an oligo that has one position hydroxymethylated. So I think we have to find a balance on finding a good oligo that's less dense in these modifications.
Can you customly synthesize?
Yes, it's expensive, but we can do that. Yeah, I think we're going to go for that. Yeah.
And my final question, a bit more general, how do you think your findings could be applied to other areas of research?
Wow, I've done many things, so I don't know. I would really like that the 5-HMC would be incorporated as a standard epigenetic data set, because now we limit everything to methylation, and that's nice. But I would also like to that research groups start to including more 5-HMC in the analysis and the discussion and putting them in the picture, in the context, and for annotating different regulatory elements, also taking into account this modification. I would say that that's the most interesting. And also to build methodologies. So nanopore, I think it's the future. So use 5-HMC to be able to train models that effectively and with high efficiency can detect this modification. I would really like that. I know that's going to happen.
Okay, thank you very much, Paula.
Thank you, Irene.
Gabriel.
Hi, Paula. So, first of you, congrats for this huge amount of work.
Thanks.
And also congratulations to the two supervisors of this super amazing thesis, and also to colleagues that have participated in here, in particular Esther Lizano, who has also contributed to this last chapter. So I think that this thesis actually, you are right, you did many things, right? And contributed significantly to many areas of our field of research in genomics. And, you know, it did both, you know, conceptually, you enlarged the framework of comparative genomics and make observations that enlarge the theory around how regulation evolves, but also at the methodological level. So you know, put attention into data generation. So it's very nice to see the interplay in your thesis, how you actually master these two aspects at the same time, which is an impressive profile for your next step, your career project, let's say. So actually, some of the things that you have been dealing with have coincided in time with the appearance, for example, for the full-length chromosome Y that you were mentioning in the text, right?
Yeah.
That was coincidental, but that means that essentially that you are fighting in the vanguard, right? Which is a more exciting place to be, but also more dangerous.
Yes.
So I will start just by mentioning a little bit of the document itself, which I found overall really very well written.
Thanks.
And concepts very well condensed and summarized. So it was a really pleasant reading through Semana Santa, you know.
Sorry about that.
No, it was actually nice. I like also the idea of attaching the review. You know, you start primate epigenomics and you start writing, and you know what, I wrote a whole review about the topic, boom, just put it there in the introduction. There's only one small, just negative, not negative, but just minor comment I would say that it would be nice to go more often. You do it, and I know you know the literature, you know, the source data for the things that you mentioned in the text, but to quote rather than the review, you know, the original source sometimes for some specific papers that I have. I think this is just really minor thing, okay? So it looks like the overarching theme or topic of this thesis is regulation. So it's regulation of DNA modifications and histone modifications.
Yes.
So I rather start just making you questions about how you see some of the topics related to DNA modifications, in this case, methylation, just to see what are your your conclusions, personal conclusions on these things. One thing is this personal curiosity, okay, but the stability of the hydroxymethylation cytosine, that you go as far in thesis as to call it the sixth nucleotide, even if it's nested to the fifth nucleotide, right? Because it has to be methylated.
Yeah.
But nevertheless, if it has its own properties, I think it's fair to call it,
Yes.
you know, just.
And I'm not the first one, just that someone said it before me.
Ah,
Okay.
It wasn't well. So how do you think, I mean, you mentioned in the thesis that this modification is stable, so it's kind of gaining its own, you know, personality in the sense that it is stable. What do you mean stable here? It means stable through time in post-mitotic cells, or it means stable across cell divisions? And if it is the second, how do you think it works? Because, I mean, you explained very well in the thesis that for methylation you have the NMT1, which essentially perpetuates the methylation state across divisions. But what about hydroxymethylation? How it is, do you think, or it is maintained through divisions? If it does, it is what you meant.
So yeah, basically what I meant by stable is that at first it was thought to just be a mere intermediate, and if you happen to see it, it's because there was a methylation, then it was going to hydroxy and forming like carboxyl, but no, it is stable. The problem is that not a lot is known about how this molecule is added, removed, or even maintained. So that's why there are so many studies about this. But it's a modification that is maintained. Actually, there are now some projects in which this modification has been studied using different human tissues, and they find this, like, it's present in most of these tissues. So it's like the modification is recurrent and it's present and it's there. But as I was saying, not a lot is known about how it is maintained.
think it's fair to call it a little bit intermediate state, because sometimes it's what it seems to be, but sometimes it's not, right? So
Yeah, that could be, yeah.
Because when we see in the brain, what we see is that if you have in the prenatal brain something is hydroxymethylated, then in the adult brain it tends to be hypomethylated, kind of predict. But if in the adult is hydroxymethylated, in the fetal is also. So at the same time, it's kind of maintained, some of them, and some of them are methylated. So
yeah, so maybe it has like a dual function, and in some cases it's more, as you were mentioning here, like a mark of where methylation will be or how it will be in the future. And in some others, maybe it has a function of its own, like its own role in regulation.
Okay. So also just another thing to reflect about this. You say, when abundance of the hydroxymethylation change, disease occurs. Could you elaborate a little bit? I mean, do you think this is a primary event in some, I'd say, brain diseases? Because you mentioned that this is highly overrepresented in brain, this kind of modification, right? So do you think it's a primary thing, or is it reactive?
So maybe I explained it wrong. What I wanted to say is that there's connection between, let's say, Alzheimer's disease and disruption in the levels that you would expect of 5-HMC. Whether if it's the cause or the consequence, I'm sorry if that's what it may look, but no, it's like there's two events, so there's something going on there, they're related. How are they related? We don't know. At least I don't know, and I think it's not fully known for most of these neurological disorders.
What is your intuition? Do you think it's a passive event, or reactive of some other higher level, first order modifications of transcription factors, or it's a primary cause, can be a primary cause in disease? Yeah.
So I think it would be, if I was to say, the first, because precisely David De Juan, they did a work in which they showed that 5-HMC, it's central in the connection of many other modifications and events. So if 5-HMC's role, it's in the center of the connection of many networks in terms of regulation, it would make sense that if you see any disturbance in, let's say, a specific protein, at the end, maybe you will see it reflected in the levels of 5-HMC. So I would go more in that direction.
Okay. Now I'm going to ask a question, even if I'm positive I would not understand the answer probably, but for the method that you developed. So I was reading the Sun et al. Reference 2021. So at some point when you mention in the thesis, you say, okay, there is this method EM, if combinations with the two enzymes, BGT plus TET2, TET1, and BGT alone before ApoBec, can help you discriminate between these two modifications, right? But then this cannot be done genome-wide. And thus, here is my method. What are the limitations? Why that method cannot be applied genome-wide?
So the main limitation is that the way that they propose the method is first you do one part of the library, then you do the modification, and then you finish the library. And that's the kit. But in the paper, they don't do that. In the paper, in order to doing this, the EM-seq, which is the standard for the methylation, the one that works, your adapters at the very beginning, that you add at the beginning, are methylated, so that after the conversion and the amination, they remain as cytosines and they can be amplified. So you cannot use these adapters for the other methodology because after the amination, they will convert into Ts, I think, so they cannot be amplified. So at this point, what they do in the Sun et al. Is they say, okay, we're going to detect it, and then in order to do the amplification, which is the limiting step, because you also have the DNA which is single-stranded, if we know the position that it's that we want to amplify, we can design the primers, and it's going to be super easy. But if you want to do it at the whole genome level, you can't. And so at first, what we wanted to do was to simply do the EM-seq methodology, but changing the combinations of the enzymes. But what happened is what I told you, that since the adapters were methylated, nothing was amplifying. So that's when we decided, okay, so what we're going to do is a library based on single-stranded DNA, because by doing that, you don't need to know the neighboring regions. And so you can simply amplify. And this is something that we were able to guess, mainly because in our lab, Esther and many others work with ancient DNA, because if not, like the standard procedure is to use double-stranded library preparation protocols. So I think it was the combination of doing this, having this problem, and being in the lab where they have this problem a lot of the time.
Yeah. Okay.
So, yeah.
Congratulations. And so, related to that, so if you now want to do a project with, say, comparative metabolomics, with, you know, for example, adding this information on the LCLs, and you had money, what method? Because I guess you can also do a long read. I mean, you said long read. You need to train the software to detect this hydroxymethylation because not yet no. But I guess you still can do these enzymatic things on the long reads.
Yeah. So we would create like short reads, and that's it. You mean put it in nanopore? Yeah.
So could you actually do the amination, and so could you do the enzymatic on the long reads?
Yeah. Yes. So the problem would be to sequence it, and the accuracies of nanopore, because at the end you want a true set, and if nanopore is not performing really well, and there are some places in the genome that is telling you it's a base that it's not because of the accuracy, you may have some problems. But other than that, I think it would be a really nice thing to do.
What would be your personal choice? What would you recommend me? Like, use my
your money.
No, use my money. And what would you recommend? Use this enzymatic that I developed. Use long reads. What would you?
If you want to do it now, I would recommend do the method here. Once we know that the controls are working, and if you can wait a little bit, with this data, be able to train models that can detect this modification in nanopore, because with nanopore at the end you have long reads and you can detect methylation, hydroxymethylation, anything. So at the end you're saving money because it's only one experiment.
Yep.
So depend on the time.
Okay. Good. I wait. Great. So I just have a couple of more questions on the Enhancer project, the project on comparative LCLs. One is probably more related to the previous part, but nevertheless I ask. So I'm curious about the weak enhancers because it's a little bit the bottom. So is there any heuristics once you have done the segmentation using HMM? Is there any heuristics a posteriori that you can use to separate? So once you have region is enhancer, like it's K27 acetylation positive and K40 negative, is there any heuristics that you can use to say this is strong, this is weak, just based on, for example, ataxic, just based on chromatin accessibility measures?
So you say only doing the 27 acetylated and then ataxic?
Yeah.
I think you could do that, but then you wouldn't be able to, well, you mean regarding on the proportion of open chromatin, I think you could do that, but yeah, that could be done. I don't feel very sure, but yes, I think you could do that.
Because I'm curious about the explanatory, the information content provided by the ataxic, that I guess it's a lot.
Yeah.
And I just want to mention, because you do partial correlations, I think you do partial correlations to explain how much of the variance in gene expression, for example, is expressed by individual marks colliding.
Yeah.
But I didn't see ataxic there, so can actually ataxic correlates more than anything else with gene expression?
I don't really know about the details of the analysis that you're saying, but in principle, it should correlate at all. So yeah, as far as as much as I would say.
But I think it's more agnostic to whether it's poised. It's just.
Yes, but for poising, poised regulatory elements, you need the 27 methylated.
Yeah.
But maybe for the weak and strong, you could differentiate. And at the end, we didn't get, or Raquel didn't get as many poised enhancers promoters. So the vast majority were either strong or weak. So yeah, maybe you would not have the poised ones.
And also related to Irene's question on the, so you restrain your analysis to the whatever is orthologous 1 to 1 to 1 to 1 to 1 across all these species, right? So, but I mean, I guess this is really positive to do it when you are essentially your universe of elements is conserved, like strong promoters or genes, because essentially you are covering the whole space, most of the space. But in regulatory elements that are like enhancers that are sometimes positionally conserved, but at the sequence level they are so degenerated, I guess that one could imagine that one design could be to consider those that are not orthologous, so you don't have an orthologous region there, then it counts as a no enhancer, right? But it's not excluded from the analysis in the sense that unless it is because of a one-to-many relationship. If it's one-to-many or many-to-many, I understand totally on your side.
Yes.
But if it is just because the region is not there, that for me is a negative. There is no enhancer here, rather than excluding the whole thing, right? I think that you would have kept a lot more enhancers that are apparently very important for the evolution, right? And not bias towards the conserved ones this way.
Yes, so yeah, you're definitely right, but I believe the regulatory elements, the orphan ones, were the ones that could not be associated to any genes. Yeah, you're right, yeah. You definitely are. I think here the main limitation was the number of replicates, which was only 2. If you would have had 5 LCLs per species, maybe that wouldn't have been a bad approach. I think here analyses were mostly very conserved because at the end you have a lot of variability with only 2 replicates. So I think they wanted to keep it safe in the sense. But yeah, sure, that would be very nice.
I will finish just a couple more things. One is, I'm just curious, so you found some repurposing of regulatory elements, so sense that they gain promoter signals from enhancers. Were those always associated to a gene, or I mean, did you actually find but unannotated genes in suddenly appearing promoters, or how it goes? I mean, there is always an annotated transcript in these new promoters.
I think those analyses were limited to those in which, but I wouldn't know the details exactly. In the ones that at least there was a gene associated in humans, because at the end the annotation is different for the different species. So I think that's limited by that.
Oh, okay.
So I would say that they were almost exclusively always, at least one human gene was there. So that would respond no to your question.
Okay. Yeah, so just my one last thing is, so you find enrichment in brain genes, which is quite striking. I mean, it was a very interesting result of the weak enhancers.
Yes.
Now, just follow my reasoning. I mean, I think that it goes this way, but just want to mention one thing. So it's that genes in the brain tend to be longer. There is some confounding effect that happens in many enrichments for genome-wide signals, right? Then if I have to select one set of enhancers here, or of regulatory elements that is more noisy in your data set, that would be of the weak enhancers, because you show in the frequency spectrum that are actually not shared by 5 species, but are rather more, the frequency spectrum is rather more, gradual, not like sharp like the promoter. So if I have to say which is the more noisy set of elements, that would be that, weak enhancers, right? They are at the threshold of detection, and also they are not very well conserved. Now, how noise would contribute to an enrichment analysis? Well, it would be essentially spread across the genome. And if that's the case, then longer elements would host more of them. So one control that has been used in other papers, approximations such as this one, is to take as a covariate the total number of regulatory elements associated to the gene. So each gene has a total number of regulatory elements. And this variable corrects a little bit the enrichment on regulatory elements. Do you think that that might
That
help?
could help. In our case, I think we were quite strict with the overrepresentation analysis. At the end, we designed one background for each of the components. So genic promoters would have a different background. Genic enhancers would have a different background. So I think if we take into account that genic enhancers, most of them are enhancers, and a lot of them are weak enhancers, I think the background already contains a lot of these super long genes. So I would say that in that sense, we really, really tried to avoid this. But yeah, what you're mentioning about the covariates would also have been nice to do. Yeah.
One more, and I finished. So
Te voy a cortar.
one more. So the final conclusion, which is that these genes are an echo of things that happen. So these enhancers that are weak are echo of something that is actually strong in the brain.
Do we say echo or like really strong, or they could be echoes?
Oh, echoes. I mean, this is in the discussion. I enjoy it very much because that is the development of the idea that comes from the paper, right? And in there, you actually expand on this. This is a cliffhanger in the
yes, it is.
total cliffhanger. And then, so did you start exploring that because the data sets are there and you actually mentioned them in the paper, Vermont paper, Canton paper, so you have human-specific elements in the brain.
Yes.
So you could.
So the truth is, we wanted to put everything in the paper from the first chapter, but this was like logistically, this was at the end of the very long process of writing this paper, the generation of the data and everything. So at the end, we find these human-specific weak enterogenic enhancers. We really wanted, I started to work with the Vermont data, data about accessibility on brain, chimpanzees, humans, everything super nice, but the results were not there yet. And so we decided to just close this chapter and say, these could be echoes, and let's see, maybe in the future we can do a proper analysis on them. Because I think it would be really nice. But then all the other chapters happened. But yeah.
Thank you.
Thanks.
Wow. Okay, this is something interesting because it's bittersweet to be the last one. Why? Because many of the technical questions that we all had are common. So sometimes you saw pieces of the questions in Gabriel that Irene had already had, right? So there is something sweet about it, which gives me the opportunity to ask you open-ended questions and try to see where you take this, right? But before that, congratulations. This was a pleasure to read, a pleasure to listen to. Congratulations to Tomas and David. This was a lot of work on everybody's count, right? And is an area that is opening very interesting directions, right? Where to go.
Yeah.
And when I look back at a lot of the results and I think about this and I think about your work, for example, on the genetic elements and the genetic roles of those elements, I think about, and I see some of the figures, the first thing that pops up to me is like, were there any unexpected results when you were doing any of these that really come to your mind and that you would like to see being worked out forward because you think that they could explain a lot of the evolution of primates that we see now?
You mean in our results?
Mm-hmm.
So yeah, basically the one about what I was just discussing with Gabriel, the human-specific weak enterogenic enhancers that have these functions associated with brain. It's like we are not expecting this. So I would really like for someone to go maybe in that direction and have a look at these elements and whether if they are really echoes as we want to like humbly suggest, or they're just at the end of the day noise that we see. Yes, I think that's one of the things that I would like to go on the direction.
How do you think that could be done? And how do you distinguish now noise from true?
So
True
the
effects.
experiments that I would do, first of all, I would go and create like, this is a lot of money, but it's okay because I'm
Doesn't
just
matter.
imagining.
Think about
So
Yep.
I would create the
Good
same
donors.
with the same species, more replicates, or at least human and other primates in brain or different cell types from brain, at least one, and see whether these weak elements that we see here are strong, are weak, are not present, are anything. Because if we don't see anything in brain, maybe this is not echo, this is just non-existent. And so if that happens, then I would go one step further, which is like, okay, let's do the same, but instead of LCLs, let's find another lineage, which is different from the one that brain derives from, like B cells. And we would then see whether what we see is the effect of the transformation of the LCLs, which we didn't talk a lot now, but these cells will transform, and that may have an effect on the biology of the cell regulation, of whether it's also true. Okay, we see these echoes in LCLs, we see these echoes in B cells. Okay, and if that's true, then I will expand to other cell types, let's say fibrous or any other cell types, to see whether these echoes are specific for LCLs, for B cells, or they are echoes everywhere of human regulatory innovations. So I think I would do that like 3-step project with a lot of money involved.
Good. And it's true. I agree with Gabriela and René that this is a very interesting breakthrough, conceptual and methodological, right? Because looking at the regulatory evolution across multiple organisms for which we don't have much functional information about what's going on, it's really hard. So how do we go now to try to expand this work and try to incorporate other organisms? What do you think are the major constraints if we wanted to continue breaking it through and taking this methodology and expanding it?
I think that the main limitation is the accessibility to non-model organisms. So at the end, here we have 2 LCLs per species, which seems like silly, like not at all, but these LCLs were super difficult to obtain. So at the end, the analyses that are done in primates are mostly from primates that are in the zoo, so their accessibility is also limited. So if we want to really expand this to other primates or other model organisms, well, not model organisms, but non-model organisms, we need to be able to access to these species. I think IPS in this case are crucial because from blood, for instance, you can differentiate to virtually any cell type. So that would be also a thing. If I had to spend a lot of money, I would do that, but I don't have money.
And what else? Like, what else do you think could be constraints on trying to expand this?
Constraint.
Like, definitely access to
Also,
material.
yeah, of course, the assemblies. We don't have assemblies for most of the species. So annotations, in order to link regulatory elements to genes, we need to know where the genes are. If there's not good assembly, we don't know where the genes are, so we end up with nothing. Also, a lot of replicates in order to see that what you see is consistent. A lot of computational power.
True thing. The other thing that is interesting is that you mentioned something, and you mentioned it in the written part of the thesis, and you repeated it here, which is true. A lot of people, when they refer to epigenetic modifications, they just refer to DNA methylation.
Yeah.
And you're saying, I mean, there are other things that are happening that are very important and are not being taken into account.
Yes.
If you were to establish a hierarchy of how important different modifications are to explain gene regulation, what would be that hierarchy? If you were to recommend somebody, it's like you need to take into account all these other things. Like, in order of relevance.
I think data on expression is really important to see what's going on. It's like when you don't have any clue, you have the expression, at least it gives you some ideas. And then to really annotate regulatory elements, I would go to 1 or 2 histone marks, or 3. Mark for enhancer, the yeah, the trimethylated for the promoter and the 27-acetylated for activity. This is the basic that I would use. Then methylation. Well, no. After the histones, I would go to ataxic and then methylation.
And now if you were to link that to biological problems, right? Like when I think about this, establishing those hierarchies, I'm thinking about what you put in your slides, which is you see these organisms that have the same genomic background and have very different tissues.
Yes.
Right? If I were to explain the differentiation of biological processes across scales, how would you put those different modifications to explain differences between species versus differences between tissue?
Sorry, I didn't follow.
So think about those processes now in a bigger context, right? It's like now I want to use all of these and I want to understand what epigenetic modifications are important, right? And looking at gene regulation that are able to differentiate tissues,
Yes.
but also species and
Yes.
important species-specific differences, right? So if I were to establish that hierarchy of modifications to explain differences between tissues versus differences between species, how would you do that?
I think they would be the same ones.
You would be the same.
Yeah, promoters and enhancers that would tell us, like, I would say that maybe promoters are not as relevant, so I would lower them in the hierarchy because at the end you have methylation, and it's more interesting to know where the enhancers are, and the enhancers will tell you about the tissue specificity. But yeah, essentially I think I would go for the same ones. Yeah.
Cool.
Reordering a little bit, but yeah, essentially, yeah.
And last, where do you think this is going now?
All this work, like of the thesis.
What do you think should be the direction in which people that wanted to follow your work and use your work as a place to start, where should people go?
I think, yes, they should try to expand to other cell lines, but basically using a lot of different histone modifications in order to annotate the regulatory element. I think that's the main limitation of most of the studies that focus on, especially those that are comparative, because in humans you have essentially everything, but the comparative ones, you lack this consistent annotation with different epigenetic marks or histone modifications. So yeah, I would just take this as the background from which to build on different tissues and different, also adding, if necessary, some other methods, technologies. So now ChIP-seq, maybe it's outdated because we have Tangakut, Kutantak, and Kutanran, and all of these cuts. So yeah, but I think it's a good start.
Well, congratulations.
Thanks a
This
lot.
was a pleasure.
Thanks.
Now we're going to ask the public to please leave us alone to deliberate.
Marina, tu tens el d'esto que m'ha llevat, això qui ho té? Ah, guai. Sí, sí, si no vaig a dir res. Que no vaig a dir res. Plorar-se.
Los íbamos a dar un ratico más afuera mientras nosotros buscábamos un cafecito, pero Gabriel pensó que no era buena idea. Bueno, Paula, el tribunal, después de ver toda la tesis escrita y tu presentación oral y evaluar todo lo que has hecho a lo largo de estos años, considera que has hecho muy buen trabajo. Y por supuesto, a partir de ahora te vamos a tener que llamar la doctora Esteller Cocala. Felicitaciones.
Gracias.
Por una tesis excelente.
Gracias.
Su tesis ha sido maravillosa y está aprobada con mención excelente.
Ahora es cuando...
Muchísimas gracias. Ahora sí pueden aplaudir.
Thank you, everyone. Now, what time is it? We'll do a pika at 1:30 in the entrance of the PREV. So, in the square, see you all there. And thank you very much for coming.